CRISPR is a topic which is highly discussed now, but it took several years for scientists to come up to this conclusion and to find out how to harness its potential and use it in other aspects of science. So let's begin with the introduction. So in 1993, Dr. Francisco Mojica, a scientist in Spain, he identified repetitive palindromic segments of DNA interspersed with other fragments of genetic material in the bacterial genomes. He named these regions as CRISPR. In 2007, a team of scientists led by Dr. Philip Horvath experimentally demonstrated Mojica's theory. Later, in 2012, Doctor Jennifer Doudna and Dr. Emmanuel Charpentier published their paper in the journal called Science, revealing that the CRISPR-Cas9 bacterial immune system could be repurposed or adopted as a gene editing tool. In 2019, the first CRISPR clinical trial began, harvesting cells from patients with sickle cell disease or SCD and editing them in vitro before infusing them back into the body, a method known as cell therapy. After the success of SCD or sickle cell disease cell therapy trials, a CRISPR treatment was injected directly into human patients for the first time in 2020, a method that is known as gene therapy. CRISPR, it stands for clustered regularly interspaced short palindromic repeats. These repetitive DNA sequences were observed in prokaryotic organisms like bacteria and archaea, with spacer DNA sequences in between the repeats that exactly matched the DNA fragments or sequences from bacteriophages or viruses that had previously infected the prokaryote. The CRISPR array is made up of an AT-rich leader sequence followed by short repeats that are separated by unique spacers. Now, CRISPR repeats range in size from 28 to 37 base pairs, and the spacers usually range from 32 to 38 base pairs. These sequences, they play a very vital role in antiviral defense system of prokaryotes, and it provides a form of acquired immunity. So here's a picture below. I don't know if it's very clear or not, but basically this shows a CRISPR array where we have repetitive sequences and they are intervened in between by the spacers. And these repetitive sequences are part of the bacterial genome, and the spacers which are there in between, these are the ones that are obtained from the viruses or the viruses which had rather previously infected the prokaryote. Now, after CRISPR, a common term which comes along with CRISPR is Cas or Cas protein. So what are Cas proteins? A major addition to the understanding of CRISPR came with Janssen's observation that the prokaryotic repeat cluster was accompanied by a set of homologous genes that make up CRISPR-associated systems or Cas, or rather called Cas genes. Four Cas genes, Cas1 to 4, were initially recognized, but now till date we have many such Cas genes and Cas proteins which have been recognized. The Cas proteins, they showed helicase and nuclease motifs. Some Cas proteins, now these are some of the Cas proteins which are discussed frequently. One among them is the Cas9, which is a term which is very commonly used while we are discussing CRISPR, like CRISPR and Cas9, they usually come together whenever we discuss this topic. So, what is Cas9, or giving a brief introduction about Cas9? The Cas9 endonuclease is a four-component system that includes two small molecules like cRNA and trans-activating CRISPR RNA or TRAC-RNA. Jennifer Doudna and Emmanuel Charpentier, they re-engineered the Cas9 endonuclease into a more manageable two-component system. So it was four. From there, they brought it down to two-component system by fusing the two RNA molecules into a single guide RNA or sgRNA that, when combined with Cas9, would find and cut the DNA target specified by the guide RNA. So Cas9 plus the guide RNA would find the DNA target that is specified by guide RNA. By manipulating the nucleotide sequence of the guide RNA, the Cas9 endonuclease would cut the DNA target specified by the guide RNA. By manipulating the nucleotide sequence of the guide RNA, the Cas9 endonuclease would find the DNA target that is specified by guide RNA. The artificial Cas9 system could be programmed to target any DNA sequence for cleavage. Next, coming to Cas12a, in 2015, the nucleus Cas12a, formerly known as CPF1, was characterized in the CRISPR CPF1 system of the bacterium Francisella novicida. Cas12a showed several key differences from Cas9, including it causes a staggered cut in the double-stranded DNA as opposed to the blunt cut that is produced by Cas9. Relying on a T-rich PAM, providing alternative targeting sites to Cas9 and requiring only a CRISPR RNA or cRNA for successful targeting. By contrast, Cas9 requires both cRNA and a track RNA. So Cas9 is requiring both cRNA and a track RNA, but the Cas12a is requiring only the cRNA. And another difference from Cas9 is that it is bringing a staggered cut in the target DNA as opposed to the blunt cut, which is usually brought about by Cas9. Is Cas13a. So this is somewhat unique. In 2016, the nucleus Cas13a, that is formerly known as C2C2 from the bacterium Leptotrichia shahi, was characterized. Cas13 is a RNA-guided RNA endonuclease, which means that it does not cleave the DNA, but only it cleaves single-stranded RNA. Now, Cas13 is guided by its cRNA to a single-stranded RNA and binds to the target. So similar to Cas12a, the Cas13a remains bound to the target and then it cleaves other single-stranded RNA molecules non-discriminately. So this collateral cleavage property has been exploited for the development of various diagnostic technologies. So these are the picturesque view of the Cas9, Cas12 and Cas13. See here, for Cas9, we have the track RNA, we have the cRNA. So they together, they are acting as the guide, which is shown by the red line. This guide is binding to the double-stranded DNA. That is my target region and will bring about cuts on both the strands. We have Cas12a, which is requiring only the cRNA, acting as the guide RNA. It doesn't require the track RNA. And for Cas13, this one is not acting on an DNA, rather it is acting on a target RNA. So the first two, that is 9 and 12, they are RNA-guided DNA endonuclease, but this third one, Cas13a, is an RNA-guided RNA endonuclease. Next, after we came to know about what is CRISPR and what is Cas, now these two will work together as CRISPR-Cas systems. Now, certain classifications are there of this CRISPR-Cas system. So the current CRISPR-Cas classification hierarchy, it includes three major types, 1, 2, and 3, and the less common but the clearly distinct type 4. The different types of CRISPR-Cas systems, or the distinction between the CRISPR-Cas systems, is based on the respective signature genes and the typical organization of the respective loci. So type 1 CRISPR-Cas systems. All the type 1 loci, they have the signature gene as Cas3. We have CRISPR, we have Cas. Now we have some different Cas proteins that are unique in each of the CRISPR-Cas systems. So for the first one, or type 1, we have the signature genus Cas3, which encodes a large protein with a helicase possessing a single-stranded DNA, stimulated ATPase activity, coupled to unwinding of DNA and RNA-DNA duplexes. Often but not always, the helicase domain is fused to an HD family domain, which has an endonuclease activity and that is involved in the cleavage of the targeted DNA. Now, each gene in this type 1 system operon is usually present in a single copy. Type 1 systems are currently divided into 6 subtypes like 1A, 1E to 1F, each of which has its own signature gene and distinct features of operon organization. Now, another thing that I've missed here is the type 1 CRISPR system. It was in this, the nucleus 3' 5' activity on the single-stranded DNAs and RNAs have also been reported for the HD domain from Methanochaldrococcus januschi. The HD domain is located at the N-terminal of the Cas3 protein or is encoded by a separate gene within the same locus as Cas3 helicase. So here Cas is having certain unique functions like endonuclease activity, helicase activity, and these things. So type 2 has a very commonly known or a unique Cas gene, that is Cas9, which includes a multi-domain protein, and it combines all the functions of effector complexes, target DNA cleavage, and everything that is essential for maturation of the CRISPR RNA or cRNA. Now, every CRISPR-Cas locus of this type, in addition to the Cas gene, it also has Cas1 and Cas2 genes. Also, in addition to the three protein coding genes, the vast majority of them encompass one or two genes for track RNA. You have genes for an RNA that is track RNA, which is an RNA that is partially homologous to cognate CRISPR. These systems, they usually use cellular RNase-3 and track RNA for processing of the pre-cRNA. Now, how this pre-cRNA is getting processed and what is happening in this CRISPR-Cas mechanism, that I will discuss next. Here I'm just briefing about the CRISPR-Cas systems and what is unique in every type. So both the nucleases are required for target DNA cleavage, and recently several crystal structures of Cas9 have been solved, including one with an artificial single guide RNA or sgRNA and a target DNA. So type 2 CRISPR-Cas systems are again currently classified into three subtypes, that is 2A, 2B, and 2C. Next, we have another type, that is type 3, and here the signature gene is Cas10, which is again a multi-domain protein, which again encodes for a multi-domain protein containing a PAM domain similar to cyclases and polymerases of the Pol B family. This protein originally was predicted to be a polymerase, but recently the structure of Cas10 has been solved, and four distinct domains again have been identified in this, where Cas10 is the large subunit of the effector complexes of type 3. Each type 3 locus also encodes other subunits of effector complexes. Type 3 CRISPR Cas systems often do not encode their own Cas1 and Cas2. This is something different for this type 3. That is, they are not encoding for their own Cas1 and Cas2 genes, but they use cRNAs produced from CRISPR arrays associated with type 1 or type 2 systems. Nevertheless, in many genomes which are lacking type 1 and type 2 system, Cas1, Cas2, and Cas6 genes are linked to a type 3 system that accordingly is predicted to be fully functional. To type 4 CRISPR-Cas systems, so similar to subtype 3A, though I have not mentioned the subtypes of type 3 again, but type 4 system is supposed to be similar to the subtype 3A. This system again lacks Cas1 and Cas2 genes, but is often not associated with CRISPR arrays. Moreover, in many bacteria, this is the only CRISPR-Cas system with no CRISPR-Cas detectable in the genome, but it has an effector complex that consists of a highly reduced large subunit CSF1, two genes for the RAM protein CSF3, and for Cas7 CSF2. In some cases, the gene has also been predicted for a smaller subunit. So for for type 4, CSF1 gene could be considered to be the signature gene for the system. Okay. So once more, I can say that is for type 1, we have the signature gene as Cas3. For type 2, we have Cas9. For type 3, we have Cas10. And for type 4, we have CSF1 gene. Key steps of CRISPR-Cas immunity. So this is the thing basically which was initially studied, and this is the mechanism which gives somewhat acquired immunity to a bacteria or acting as its antiviral defense machinery. And this phenomena has been exploited and utilized in different aspects of science, which I will discuss later on. So firstly, coming to how is this working, CRISPR-Cas immunity in bacteria. So the first step here is the adaptation, which leads to the insertion of new spacers in the CRISPR locus. So this figure very clearly shows this brown colored bacteriophage. It is affecting the bacterial cell. So this is the first exposure. We have the viral DNA that is getting into the cell. Now this viral DNA is getting incorporated into the CRISPR sequence or array of the bacteria, and they are being held by Cas1 and Cas2. So this step is called the acquisition, or it's also called the adaptation stage. And the next one is the second stage. The system gets ready for action by expressing the Cas genes. So this entire part, that is the primary transcript, will now be expressed and transcribing the CRISPR into a long precursor CRISPR RNA or the pre-cRNA. See, this one shows the pre-cRNA. The CRISPR RNA track RNA duplex is cleaved off the target pre-cRNA. Now, the pre-cRNA is subsequently processed into a mature cRNA by Cas proteins and other accessory factors. In the third or the last stage, which is called the interference, here we can see this entire CRISPR-Cas machinery is ready in the cell. Now the cell is ready to fight back any viral infections that will come up to the bacterial cell for the next parts of their lifespan. So in the third and the last stage, interference, where the target nucleic acid is recognized and it is destroyed by the combined action of cRNA and Cas protein. See, this is the last part. Here we have the target DNA, obviously. We have the viral DNA here. And the second time, this is the second exposure, the bacteria is affecting the cell. And we have the CRISPR-Cas machinery ready. Here it is with Cas9, and it is degrading the cell, the viral sequences. So genotoxic double-stranded cleavage. So first, three basic steps of this immunity. The first one is the adaptation stage. The second one is the production of CRISPR RNA or cRNA. And the third one is the DNA hybridization and cleavage of the viral genome. Now, this is the CRISPR mechanism. Just like I have mentioned previously, this is in a much more detailed format. So when a bacterium, so here very well we can see this brown color part is the Cas9. Along with this, we have the guide RNA or the gRNA. This is the double-stranded sequence. And this guide RNA will bind to the part that is to a particular part of the to the viral sequence, like where it is compatible, obviously it will bind to that part. So how is this working? When a bacterium is infected by a virus, it uses a Cas nuclease to snip off a piece of viral DNA that is known as the protospacer. See, we have protospacers here. This fragment is stored in the bacterial genome with fragments from other viruses that have previously infected the cell. So this is a form of immune memory which allows the cell to remember, recognize, and clear off infections. Upon reinfection with the same virus, the bacteria can recognize and destroy it with Cas9. And Cas9 requires, as I've told previously, cRNA, that is CRISPR RNA, and a TRAC RNA or transactivating RNA. The cRNA is complementary to the viral spacer that was stored after the original infection. See, this is binding to the viral spacer, while the TRAC RNA serves as a scaffold. Now, these two RNAs, they form a complex that is known as the guide RNA or the gRNA here. So before cutting, the Cas9 will obviously act as a search tool. That is, it will check the viral DNA for the protospacer adjacent motif or PAM. See, here we have the PAM or the protospacer adjacent motif. So when the Cas9, it recognizes the PAM, so PAM is present downstream of the target site. When it recognizes the PAM, Cas9, it checks the region now upstream if it locates the target that is provided by the gRNA. And then it will create a double-stranded break or DSB. DSBs incapacitate the virus because viruses lack their own DNA repair mechanisms. Now, CRISPR-Cas9 gene editing works by creating double-stranded breaks in the DNA, and then it takes advantage of the cellular DNA repair mechanisms. While there are several repair mechanisms, the key ones that are used are NHEJ, that is non-homologous end joining, and homology direct repair or HDR. Dr. Doudna and Dr. Charpentier also revealed that if Cas9 is given a different guide RNA sequence, it could still be used to create cuts at a range of target sites or location in the organism's genome, relying on the presence of correct PAM sequence. So it needs to recognize first the PAM sequence, which is a part of the viral genome, and then the guide RNA will bind to the target sequence, and then it will bring about a double-stranded cut. So even if you are making different guide RNA, you can cut at several sites of the target genome. But again, that has to be done in the presence of the correct PAM sequence. PAM is adjacent motif. Now next, some factors that are affecting the CRISPR-Cas9 system. We have target DNA site selection and sgRNA design. These are the things that are essential. We have to select a proper target DNA and single guide RNA. Like one report, it suggests that the CRISPR-Cas9 may be less specific than ZFNs or TALENs due to the relatively shorter targeting sequence. This contrasts with many reports that show no detectable off-target cleavage from CRISPR-Cas9 editing, with off-target effects being guide RNA specific. Okay, so this is another drawback which we have to keep in mind while we are carrying out this experiment. The rational design of sgRNA has therefore been the subject of a significant body of work. Now, there are many computational tools and software packages that are available to facilitate your single guide RNA or sgRNA design. However, caution is still needed as some authors show the evidence of algorithmic overfitting. So, the incidence of HDR-mediated DNA repair from DSBs is typically low in mammalian cells. Even in the presence of donor template DNA, NHEJ or non-homologous end joining is the more frequent repair mechanism that is observed from CRISPR-Cas9 editing. Now, several approaches have emerged to increase the HDR efficiency and suppress the NHEJ, including use of small molecular inhibitors of NHEJ, gene silencing, cell cycle synchronization, and use of cell cycles deficient in NHEJ complexes. So one of the most commonly used inhibitor is SCR7, which targets the NHEJ component DNA ligase 4 and has been reported to increase the efficiency of HDR from Cas9 editing by up to 19-fold times. So first to our target DNA site selection and sgRNA design. First thing to be kept in mind, the drawbacks have also to be kept in mind. The next one is the efficiency and the incidence of HDR. We usually try to avoid HDR and go for NHEJ. But again, we see that the incidence of HDR mediated DNA repair is more in the mammalian cells. So we need to avoid that HDR and we need to go for NHEJ. And we have some components which are acting as inhibitors. One is the Cas9 activity. Cas9, the gene or the protein is extremely influential in this CRISPR-Cas systems. And for gene editing in eukaryotic organisms, Cas9 must translocate into the nucleus. In these systems, the nuclear location signal or NLS is connected to the Cas9 protein, increasing access to NLS by adding a 32 amino acid spacer between the NLS and Cas9 was shown to increase DNA cleavage activity. So increasing the relative concentration of sgRNA to Cas9 protein was also shown in increase on target cutting activity, presumably by ensuring that all Cas9 proteins formed the active ribonucleoprotein complex. However, excessive sgRNA was also shown to increase off-target effects. So Cas9 concentration, Cas9 effect was also to be kept in mind while we are carrying out this activity. Okay, finally, in comparison with other enzymes, activity of Cas9 is quite low. And once bound to the target DNA, displacement from the DNA strand, even after the DSB formation or double-stranded break formation, is challenging. Thus, Cas9 is less like a catalytic enzyme and more like a single-shot actuator. So while this characteristic may be somewhat useful in some instances, like gene activation or inhibition, or short-lived activity for gene editing, with lower off-target effects, it may be undesirable for other applications where the catalytic activity is useful. So you have some advantages, some disadvantages. So keeping in mind our target experiment, we need to choose accordingly. Next, we have off-target cutting. So in addition to the rational design of sgRNA, we need to improve its specificity and reduce off-target cutting. That is, we need to specify our target only and we do not want off-target cuts. We only want on-target cuts. And reduce off-target cutting have resulted in the design of mutant Cas9 system. So while Cas9 itself does not cause off-target effects, they exist solely due to the sgRNA. Improvement to the Cas9 protein can limit these effects nonetheless. So one mutant system disrupts the Cas9 protein so that it introduces only single-stranded RNA nicks. The nickase is then used as a pair with one Cas9 to bind to the forward DNA sequence and another Cas9 binding to the reverse DNA sequence, flanking the target site. Only when binding is in configuration is a double-stranded break formed through cooperative nicks. So again, off-target sequence is something that has to be kept into mind to ensure the specificity of this CRISPR-Cas9 mechanism. Under the factors that are responsible or may affect this system is the CRISPR delivery system. See, this delivery entire process, it can be broken into two major categories. That is the cargo and the delivery vehicle. So regarding the CRISPR-Cas9 cargos, we have certain approaches. Delivery vehicle used will often dictate which of these three cargos can be packed and whether the system is usable in vitro or in vivo. As an example, Cas9 is positively charged, but oligonucleotides and Cas9 sgRNA RNP, they are negatively charged. Additionally, considerations for how tightly controlled the overall concentration of Cas9 is must also be made. By introducing Cas9 DNA instead of protein, it becomes more difficult to ascertain precisely how many functional Cas9 units are present in the system at any given point of time. So we need to like tally and see which cargoes are compatible in which delivery vehicles. So some of the vehicles that are usually used are classified under physical delivery, viral delivery, and viral vectors and non-viral vectors. So the viral delivery vectors are usually the engineered AAV, adeno-associated virus, adenovirus, and lentivirus. Especially for in vivo work, viral vectors have found favor and are the most common CRISPR-Cas9 delivery vectors. The non-viral vector is not as prominent as the viral-based delivery. However, non-viral, they possess several advantages over the viral ones, and they are still a process of ongoing research. These include the lipid nanoparticles, the CPP, cell-penetrating peptides, and gold nanoparticles. We have additionally many other delivery technologies that are available suitable for the CRISPR-Cas9 systems. This is a diagrammatic representation of the CRISPR delivery system. Now, mechanism of CRISPR genome editing. So the CRISPR system is used for gene knockout. So gene knockouts are used in a range of research areas, including functional genomics, pathway analysis, drug discovery, and screening, disease modeling. Using multiple guide RNA that target various regions of the gene ensures high efficiency gene knockout, a method that is gaining popularity. We also have gene knock-in along with that. So in presence of a DSB induced by Cas9, cells can also repair themselves via HDR, and this pathway offers an opportunity for researchers to insert a new piece of DNA or an entire gene. So this is called gene knock-in. And to achieve this, you need to have a DNA template for repair that is known as a donor template. Donor template consists of sequence or gene of interest flanked by regions of homology that match the area on either side of the cut. The donor is then delivered to the cells being engineered along with other editing components of Cas9 and sgRNA. So compared with gene knockout, gene knock-ins are more challenging. This is because the HDR is less common repair pathway than NHEJ, only occurring at certain stages of the cell cycle. The low frequency of HDR typically results in low knock-in efficiencies. However, scientists have been creating methods to overcome this obstacle. So we have gene knock-in and knockout with the help of CRISPR. So this is the thing. We have a targeted locus. We have the CRISPR machinery acting over here. We have double-stranded breaks. Then we have two repair machinery. One is the NHEJ, shown by these dark black arrows. Another one is the homologous recombination or HDR. To NHEJC, we have a gene knockout over here. And if you want to have a gene knock-in, we have our chromosome or the gene where a particular part has been knocked out. We have the donor DNA. This donor DNA will be inserted into my target sequence. So this is the gene knock-in followed by a selection. This is an example of target gene knockout followed by a selection marker knock-in. Next, we have CRISPR-A and CRISPR-I. So the CRISPR-Cas is used to delete or insert genes, but again, there are slight modifications to this system, and it can also be used to regulate the expression of genes, like CRISPR-A with activation of certain genes and I with interference of certain genes. That is, CRISPR-A is upregulating the expression of certain gene and I is downregulating the expression of certain genes. Of these technologies, they function by using an engineered variant of Cas9, which is called catalytically dead Cas9 or dCas9, with transcriptional effectors to modulate the target gene expression. So again, these modulators have many research applications, including developmental biology, infectious disease, disease progression, functional genomics, screening for genetic elements that mediate drug resistance. Next, so I'll just show the picture of this CRISPR RA and A and I. So this is the normal CRISPR, the first line. The second diagram or the second line here shows the CRISPR-I. So this is an RNAP block. This is down regulating the expression. The next one is CRISPR-A. This is up regulating the expression. One is base editing and prime editing. This is somewhat very unique and important. So some of the recently developed CRISPR methods are base and prime editing. So they work on the same principle, but on a more precise scale, including SNPs or single nucleotide substitutions. Importantly, the base editing and prime editing do not induce DSBs in the target DNA. Okay? So the base editing, it uses a single catalytically dead Cas9 or dCas9 or a nicase Cas9, that is N-Cas9. dCas9 is capable of cutting the DNA, while N-Cas9 produces nicks or single-stranded breaks in the DNA. Using of prime editing. In prime editing, you have N-Cas9 that is fused to an engineered reverse transcriptase and a prime editing guide RNA or a PEG RNA. The PEG RNA has two sections, one which guides you to the region of interest and the second one which contains the desired substitutions for repair after the single-stranded cut has been generated. After one strand has been altered by the prime editor, the complementary strand will also be corrected. That is, an additional gRNA and NCAS will create a nick in the other strand and it will be repaired using the previous machinery, using the previous strand as the template. So prime editing is predicted to be capable of treating 89% of genetic mutations in humans. So this picture below, it shows, this first one is a DSB mediated by HDR. The second one shows base editing and the third one here shows prime editing. We have a PEG RNA, we have a reverse transcriptase. So these two are specifically directed for single nucleotide changes or substitution, where base editing is not capable for all the kinds of single nucleotide changes, but prime editing is capable to handle those specific changes. Now CRISPR protocol. So after we came to know about what are the details of this CRISPR for Cas and how these two systems work and what are the types and subtypes. Now we need to design our experiment. So after becoming familiar with the basics of CRISPR, we are designing experiment using these main steps. So first one is guide RNA design. This is one of the most crucial steps. The choice of guide RNA will very much depend on the type of edit you require, whether it is a gene knockout or a gene knock-in, base or prime editing, CRISPR interference or CRISPR activation. Then choosing the right Cas variant. This is also very important for the success of our experiment. 9 was the first and the most commonly used Cas nuclease, originally isolated from Bacterium Streptococcus pyogenes. However, there are many differently naturally occurring and engineered Cas variants available for different purposes. This is the second one. The third one is the transfection protocol. So there is no universal transfection protocol for all experiments because delivery of editing components, it depends on the cell and the organism that you are targeting, as well as the type of editing cargo that is being delivered. So some researchers have determined that the best method for introducing gene editing components into majority of cell types involves nucleofection of RNP complexes consisting of chemically modified synthetic single guide RNA or sgRNA and purified recombinant Cas9. Okay, so transfection protocol is very essential. Now, analyzing your experiment after you have transfected, the last step in evaluating your experiment is picking an analytical tool to confirm that the editing was successful. Like every other step, this must be tailored to the experiment. The gold standard used to use an NGS or next generation sequencing. However, it's not always possible in every lab's budget, especially if we need to sequence frequently or we have many samples to run. In case we have certain companies which are offering such kinds of edits, where one such is Synthego's Interference of CRISPR Edits or ICE. This is an online tool for analyzing the Sanger sequencing data, which is obviously cheaper than an NGS, user-friendly, open source, and highly accurate, which with results that are comparable to NGS. So these are the four basic steps for designing a CRISPR experiment or to apply this CRISPR in our practical works. Now, whatever we have discussed is everything about gene editing or genome editing, whatever we might say. Now, CRISPR has something to do beyond genome editing. I've just discussed few steps, though we have many, like many applications are there, and The research with CRISPR is a very vast one. Uh, Still, I've mentioned some of them. So, inducible CRISPR systems were also developed. A photoactivated Cas9 was generated from a split of Cas9 fragments in photoinducible dimerization domains. In response to blue light, the CRISPR-Cas9 system performed gene sequence modification. Editing activity was extinguished by removing the light source. A similar blue light activating system was developed for epigenetic gene activator CRISPR system in a particular paper by Hilton, which I've just mentioned. The chemically induced CRISPRs have also been created. Now, the another one is the specific DNA binding function of CRISPR has also been repurposed to detect the location of genes. Till date, we were like editing genes, gene knockout, gene knock-in, or uplifting their expression or down-regulating their expression. Now, a particular interesting part of CRISPR that has been found is to detect the location of genes within undisturbed nuclei of fixed cells in living human cells, termed as Cas-FISH. These Cas9 proteins were labeled with differently colored fluorophores and coordinated to a specific sgRNA. This allowed for multicolored detection of specific genomic loci with high spatial resolution and assessment of DNA compaction. Unlike the traditional fluorescent in situ hybridization or FISH, this technique avoided the need of heat treatment and chemicals that can distort the drug delivery 1239 natural organization of the nucleus.